In this video, we are going to explore the diversity of fungi. So this is a phylogenetic tree reminding us how the fungi and animals actually share a more recent common ancestor than each of them do with the plants. So we are more closely related to fungi, in fact, in terms of coverings, exoskeletons like chitin, fungi and animals. Many animals share this type of carbohydrate. So for example, in the exoskeleton of arthropods, there's chitin. And in the cell walls of fungi, there's also chitin. The sister taxon of fungi, as we can see here, are the chytrids. Okay, so that's the first one that we are going to see in this particular presentation. Okay, so the four groups of fungi are the zygomycetes or the zygomycota with 1,000 species. Then we have the glomeromycetes, and then the ascomycetes, which has the most number of species, and the ever so familiar basidiomycetes, where our mushrooms belong to. Okay, so the chytrids or phylum Chytridiomycota are found in very varied environments, from terrestrial to aquatic environments, and even in hydrothermal vents. Their main characteristic is that their spores, if you can remember the spores in plants, the chytrids spores are flagellated. Okay, so there are flagella for the spores. So these spores are known as the zoospores. Zoo meaning animal because they can move. Okay, like animals. Animals, we usually associate animal characteristics to movement. So they can be decomposers, parasites, or mutualists. And it's through molecular genetics that we actually found out that they are a sister taxon to fungi. So this is where the common ancestor is for fungi and chytrids. So here we have the chytrids, and the sister taxon is the group of fungi. So this is a picture of a flagellated spore of a chytrid. So again, we call this a zoospore. So that's a characteristic that differentiates them from, say, protists or from plants. So the first group of fungi are the zygomycetes, also known as phylum Zygomycota. So this is where the common bread mold, the black bread mold, belongs to. So if you're familiar with bread molds, most likely that type of bread mold is this Rhizopus stolonifer, which belongs to Zygomycota. So their hyphae, as we mentioned in the previous video, there are two types. Zygomycetes have sinusitic hyphae, meaning to say there are no septa. And they have a sexual sporangia which produce haploid spores. They are named so zygomycetes because they produce a sexual structure called the zygospore, okay, or zygosporangia. So this is where karyogamy, or the unison of nuclei, and where meiosis happens in this type of fungi. So this is the life cycle of a black mold, Rhizopus. So we can see here the fruiting bodies. So these are the fruiting bodies of Rhizopus. Okay, you can see the spores here when the spores are released. Okay, and when they land onto moist surfaces or maybe on bread, they can actually grow and release mycelia, so the thread-like structures. Okay, and if male and female spores, we call them mating types, see each other, you can see in this portion the red and the blue nuclei unite. Okay, so that's because we have a mating type, a negative mating type here on top, and a positive mating type here at the bottom. Okay, when the two unite, they actually are going to form this structure in the middle between them. So here we can see the two mating types. So this one could be the negative mating type, and then this one could be the positive mating type. And then in the middle, you have your zygosporangium, okay, or the zygospore. So when the zygospore detaches, okay, the nuclei will be united, okay, in the zygospore, and they will undergo meiosis. Okay, and then after meiosis, this particular fruiting body is going to emerge from the zygospore and then releases spores again, which may land onto favorable surfaces, and the life cycle of the fungus continues. So we see here in purple a short span of time where the fungus is heterokaryotic, meaning to say the nuclei, as we see in this illustration, are mixed together. There's no boundary between them. And we see a short portion here after karyogamy, just before meiosis, where the fungus is in the diploid stage. So this is the only diploid stage. Okay, so this is the life cycle of the bread mold, black bread mold. Next, we have the not so common phylum of fungi, phylum Glomeromycota, or the Glomeromycetes. So it's stated here they were once considered part of the zygomycetes, but molecular analysis proved that they are separate from the zygomycetes. So nearly all species form arbuscular mycorrhizae. So that's the reason why we don't really see these types of fungi, because they usually associate themselves with the roots of plants as mycorrhizae. Okay, so this is how it would look like inside a plant cell in the roots. And then our next phylum of fungi are the ascomycetes, or the ascomycota. These are also known as the sac fungi because their spores, their sexual spores, are stored in sac-like structures, so we call asci, okay, which is contained in the fruiting bodies, which are known as ascocarps. Okay, so that's why they're known as sac fungi. So examples would be morels, which look like mushrooms, okay, but they have a distinctly different appearance. And then we even have unicellular ascomycetes, so which are also known as yeasts. So by now you should put in mind that when we say yeast, we are not only talking about the yeast used in bread making. Any unicellular fungus that is not thread-like in structure can actually be considered as a yeast. Okay, so back to the phylogeny. So we can see here that the zygomycetes actually parted ways from the rest of the fungi early on, and then followed by the glomeromycetes, and then the ascomycetes, which we just talked about, is a sister taxon to the basidiomycetes, our common mushrooms. So here's a moral. This one is an ascomycete, okay, belonging to Ascomycota, and even the truffles, okay, which are prized flavorings in food, are also part of the Ascomycota phylum. Okay, so ascomycetes produce asexually as well. They also reproduce asexually through the use of spores we call conidia. Okay, conidia are produced at the tips of specialized hyphae called conidiophores, and they may also participate in sexual reproduction by fusing with a hyphae of a mycelium of a different mating type. So again, let's pause here. The sexual and asexual reproduction of fungi is quite different from how we see sexual and asexual reproduction in plants and animals. I think so far you haven't heard of the terms sperm and egg, because we only refer to male and female fungus as mating types. Okay, so these mating types have their own hyphae, negative and positive, and they can do their own asexual reproduction, but when a negative hypha meets a positive hypha, they can unite together, and that's already considered sexual reproduction. So here's the life cycle of a sac fungus, an ascomycete. Okay, so again, here is the asexual loop, wherein a conidiophore that grows from the mycelium. Okay, here are the conidiophores. Okay, so those circular structures at the tip of the branching mycelium are conidiophores. When they detach, okay, we have here the conidia, and the conidia can land onto favorable surfaces, and they may also grow into mycelia of negative and positive mating types. And if a negative and a positive mating type mycelium fuse, okay, plasmogamy happens. Okay, and in plasmogamy, there's a structure called an ascus, where the two nuclei, as you can see here in blue and in red, mix together. So it's still dikaryotic. Okay, that's the equivalent of heterokaryotic, dikaryotic in this case. And then later on, the two nuclei fuse. As you can see here, there's only one nucleus now inside the ascus. Okay, so that's the process of karyogamy. And this is the only stage, the one in pink, that's diploid or 2N. And then meiosis happens, okay, producing 4 haploid nuclei inside of the ascus. Okay, so here's a picture of an ascus. And then the ascus is actually stored in bundles inside the fruiting body, which looks like a mushroom, okay, also known as the ascocarp. And when the asci, plural for ascus, break, those ascospores that are haploid are released, and when they land onto favorable surfaces, they again produce hyphae and eventually mycelia, and the cycle can repeat itself again. And then lastly, we have the phylum Basidiomycota. So this is where our edible mushrooms belong to, even puffballs, and even shelf fungi, those bracket fungi that grow at the barks of the tree that look like shelves, literally. Okay, they are known as basidiomycetes because they are known to form club-like structures known as basidia or basidium. Okay. So because of that, they're also known as club fungi. So again, the basidiomycetes are the sister taxon to the ascomycetes, as we see here. So here are examples of basidiomycetes. We have the puffball here. Then we have the shelf fungi. I'm sure you've seen all of these. Then we have a very intricate fungus here, the maiden veil fungus. And of course, the mushrooms and toadstools. So the fruiting body of basidiomycetes are known as the basidiocarps. So if you've eaten mushroom in can, or you've cooked mushroom, mix it with your food, you're actually eating basidiocarps. Okay? So if you love pancit and then you have those tenga ng daga mushrooms, quote-unquote mushrooms, those actually belong to Ascomycota, so those are known as ascocarps. So here's the life cycle of the basidiomycetes. So again, we start here. The male and the female mating types fuse. Okay, so the two hyphae fuse in a process called plasmogamy. Okay, and when they fuse, they go through a stage called or described as dikaryotic because the nuclei remain separate. Okay, and that actually happens here in the basidiocarp. Okay, and then eventually the nuclei fuse in structures we call as basidia, which are found underneath the mushroom cap. So that's where the gills are. Okay, as you can see here, where the gills are. So there are club-like structures, and in each of them there are two nuclei which then fuse in karyogamy. So now you have a diploid basidia. You have diploid basidia. And then after that, meiosis will happen, producing haploid spores contained or attached to the basidium. So as you can see in this picture, we have the basidium, and then we have here the haploid spores, which can then detach and fall and land on favorable surfaces, and then grow into mycelia again. And then there will be a positive and a negative mating type, and they will fuse again in plasmogamy, and the cycle repeats itself. So sometimes during after a thunderstorm, after intense lightning, you would actually notice that overnight mushrooms will grow in your garden or in your field. And that's because lightning is able to fix the elemental nitrogen in air into nitrates, which is a nutrient that not only plants love but also fungi like these mushrooms. And this is called a fairy ring, and you might be tempted to think that fairies actually play here at night, but no, I'm sorry to say that's not true. The shape of this is like a ring because this actually started as a mycelium here at the center. Okay, now as the mycelium grows, it becomes wider and wider in diameter. Okay, so we just don't see it because it's underground, it's incorporated in the soil. So let's say there's a thunderstorm and it's already up to this level. When there's a thunderstorm, the mycelium produces fruiting bodies on the edges. So here at the edges, mushrooms will grow. And because the pattern of the mycelium growth is circular, the pattern of the mushroom growth is also in a ring-like structure we call the fairy ring. And then we have this mutualistic association between fungi and green algae we call lichens. Okay, And the fungi involved here are often the sac fungi or the ascomycetes. And here's how lichens look like. Okay, so some of them look like liverworts, okay, from your bryophytes in Kingdom Plantae. So please don't confuse them, okay. These are associations between fungi and algae. No plants involved here. And lichens are believed to be good indicators of air quality. The leafier a lichen is, the cleaner the air in that area is. So if you go to remote areas in Bukidnon, or if you climb a mountain like Mount Kitanglad, you will see many leafy lichens growing on the barks of the tree because the air quality in those areas are really good. But here in Xavier, if you've checked the trees, they're growing. I think there's a tree there at the Lucas Hall area. There are trees there with lichens growing on their barks. The lichens there are crusty, like this one. So they're crusty because the air quality, unfortunately, here is not as good as that of rural areas, for example. So good air quality indicator, those are lichens. And this is a microscopic structure of a lichen. You can see how the fungi hyphae provide structure for the algal cells to anchor on. Okay, so the fungi provide structure while the algal cells make food, and the fungi also get some of that food. So that's how the mutualistic relationship works here. So we already know that fungi are decomposers, but many of them are also parasites to plants and to animals. So they're pathogens. So if you happen to have fungi, parasitic fungi on your body, you have a type of mycosis. Okay, a disease brought about by fungus. So for example, athlete's foot, okay, or thrush in the tongue. And then in plants, fungi are also pests. They can kill crops like these corn smuts and these ergots growing on rye. But we also have practical uses of fungi. So many species are of practical use. So of course the penicillium, I think, was the first source of antibiotic. And then there are many other fungi, mold fungi, that are also sources of antibiotics, modern antibiotics right now. And of course, we use them for food, like mushrooms, and we use them to brew our alcohols, like in beer, for example. Okay, so fungi also have many practical uses to us humans.